Okay, have we ever been back? 2 o'clock, 2:02. We're going to finish at 3:15. Okay, I want you guys one by one to tell me one thing that you've learned in last 6 days. One word. Okay? Goal is to ensure you guys are able to briefly describe, right? Remember when you have a startup company, you get 1 minute for an elevator pitch. How many of you have heard of that? We're able to summarize one word. Okay? Come here in front, guys. Who's people who are here? Just please come on the right up front here. This is your you owe me this for last 6 days. One word that you think you have learned. Okay? Or two. It's okay. I'll give you two. Cool? Okay. Let's start on the right side. Okay, your one word is over. So you summarize one thing. Genotype and phenotype is different. Right? That's what you said? First sentence? Okay, that's your learning. One, right? I just want one word summary. I'm just trying to get you guys to express yourself. Okay? All right, you can sit down. One word summary, guys. Okay? Next one. We want to keep it quick, short. I want it done for 5 minutes. One word that you've learned in last 6 days. One thing that you've learned. Quick, right? One word, one sentence if you guys are having trouble with one word. Great, thank you. Next one. Great, great. Next one. Here. We're going back and forth column wise. Okay, great. Next one. Okay, next one. Great. Next one. Wonderful. Next one. Great. Next one. Okay, next one. Good. Next one. Great. Wonderful. Thank you. Great. Thank you. Wonderful. Next one. Great. Next one. Great. Thank you. Next. Okay. Next. You learned it today. Okay. Next. Great. सर आपको एक पीडीएफ भेजा और लगाएंगे. Great. Thank you. Great. Next one.
Even a small environment change can induce an epigenetic change.
Wonderful.
Cancer is really fast.
Great. Next one.
Changes everything in body. Just techniques.
Lovely. Next.
Next generation sequencing and chip sequences and genome.
Okay. बहुत बढ़िया. Next one. Okay. Very nice. Next. Transcription is translating histone code. Okay, great. Last. Okay, great. It's so nice to hear different things that you guys have spoke of, right? Wonderful. It should teach you in your own brains how to summarize something in 1 second. What mattered to you the most, if whatever came to your brain first probably was the best learning that mattered to you, your brain the most. We have subconscious brains, and subconscious brains are amazing, and they actually drive you to do things without you even knowing, right? It's not a thing. So that's why I wanted to do an impromptu exercise for you guys to tell me, what did you learn in the last 6 days? I hope that was very impactful, right? Hopefully you'll keep that in your mind for a long time, and hopefully this continues to evolve you, right? The last hour for me with you guys. Okay. All right. ये नया वाला कि पुराना वाला? Okay, good. Thank you. Remember this picture. Okay, what is this? Corn. Why? What corn? What is with corn? What's up with black corn and the yellow corns? Remember these pictures, right? Pictures are impactful. We are a visual people. Why am I showing you the picture? Because the phenotype I showed you is because of transposable elements. And I told few minutes about transposable elements because I think it's absolutely crucial about our genomes, our epigenomes, and how we become who we are. It tells you about evolution, who we become because of evolution, right? How viral sequences have integrated in our genome. We've taken them up, used them, and adapted with it, right? And how they impact our diseases. Cool, right? I love it. Transposable elements make a majority of your genomes of one category, if you will, beyond genes. Well, you're not even before genes, okay? So one category that will make is that. What is a transposable element? Very nice. A piece of DNA that has ability to move and integrate itself into the genome is a transposable element, as the name suggests. So activated transposons copies, carry and move itself within the DNA and gene activity. So they move around. They're normally silent. They can move around under specific stress conditions, epigenetics, environment, stress, right? And can then lead to change in genomes, change in transcriptome, and a beautiful flower becoming a demon, right? Right? Or the cons getting different phenotypes. Barbara McClintock was the person who discovered this many, many years ago, right? Cool? All right. There are 3 types of transposons, since I have to be didactic in nature here, right? Which you can read. I mean, I just Googled it and I found the slide. So you guys, I'm going to be very upfront and honest with you how I made my slides, right? My job is to structure the presentation for you. Slides somebody else can make. That's not my main problem. Okay, there are 3 types. Retroposons are ones which are RNA based, right? Which actually make an mRNA. Cool? Genomic DNA, transcription, mRNA, they can reverse transcribe and then they get integrated. There are non-LTR retroposons, they're also mRNA based, okay? But they're not based on the LTR. LTR are the repeat elements that are found in 2 ends, okay, of each transposon, and those LTRs allow the recombination to happen because of the same sequence homology. You have heard of recombination? Okay, good. And then there are other kinds, mRNA based, that actually get prime, that become prime reverse transcription based on like use of their poly A tail. I'll talk about that in the next few slides. And then finally, DNA transposons that just integrate the DNA, no RNA involved. All right, cool. 3 different kinds. Let's see the next slide. Lines, I just talk about the middle ones because lines and signs, because I got the slides from Arpan. So I must thank Arpan in the last lecture. He was amazing. He was I was very thankful to him. But you guys can go watch his YouTube videos, okay? Thank you. Thanks to him. Thanks to Debu for giving me the slides as well. And thanks to all those people on the Google who put their slides up so I can just put it up and find it and I don't have to do all the work. Okay, cool. The lines are probably the most important retroposons in our genomes. There are a large number of them. So if you look for just transposable elements, I think probably the largest category is lines and signs. Okay, and lines are the ones, they contain 2 ORFs, as you can see. The retroposons of the other kinds also contain GAC, POL, and REV, which is essentially make their packaging, etc. normally. So, but this one has ORFs that contain they stand for long interposed nuclear element because they're large in size. I think 2,800 base pairs or so. The short ones are shorter in size and a few hundred base pairs. I don't remember the exact size. It might be here. So one of the ORFs contains for an RNA binding protein. So they're going to have an RNA. They're going to code for an RNA binding protein. What does RNA binding protein mean? Protein that binds RNA. It binds to itself, its own RNA, so that it can transpose it. Okay? That's RNA binding protein. And then encodes a protein with endonuclease and reverse transcriptase activity. What does endonuclease mean? Inside the DNA, can cut it. And then reverse transcriptases, right? So it's going to it has a poly A tail, it's going to reverse transcriptase itself, make its own cDNA. Yeah, you guys are smart. I made you smart in 6 days. You were born smart, guys. Thank you. All right. So signs don't have any of this, so they depend on lines for doing this. So they essentially have, oh, I'm going too fast. I don't have enough slides. So I will end before 1 hour. I'm supposed to go for 1 hour. Okay. But we'll have fun. Okay. Cellular RNA transcribes integrated line sequences. So I just told you this earlier, the line mRNA is generated that binds the DNA, the RNA binding part of it binds to it, okay? And because it has a poly A, it can look for sequences that has DT in it, right? Because A is complementary to T. So essentially, if endonucleoside cuts it, the TTT is exposed, can put its own, AAA, that's priming it now. Going back to the first strand synthesis, as you can see on the slide, it's very easy, and that gets integrated then the second strand synthesis, of course, first and second, then DNA joining and repair goes boom back. I got a new sequence inside my genome. How cool is that? They're self-engineering, they're self-genome engineers. They engineer the genome by themselves wherever they want to go once they're expressed. Epigenetics represses them, right? So they are mobile elements. Mobile elements should not be hopping around in the genomes, otherwise they'll create DNA breaks, they'll create these problems, they'll reactivate tumor suppressor genes, they'll do all kinds of shit in your cell, right? You don't want them to do that. You don't want your cells to go crazy. So we need to keep them suppressed. In cancers, though, they get up a lot, lots of times. Okay, and when they get up, what happens? They make mRNAs, right? They make mRNA? No, they make RNAs, of course. Okay. I told you guys this earlier. I don't have a slide for it, and it's a very important concept because we talked about earlier in the previous lecture about epigenomic changes not correlating with transcription, right? I talked about that. So what we need to figure out is what do they do, right? What do the epigenomic changes do? This kind of epigenomic changes will, well, they correlate with transcription. They'll make RNA. We talked about protein early on a lot. This will not make a lot of protein or whatever proteins. These RNAs that are made actually induce a signaling called C-gas-sting signaling. I think I discussed this earlier to you guys too. C-gas-sting signaling is very, very important. It is important in the immune system, and I think I was talking to Dr. Ravi, who's going to teach you tomorrow. He's going to teach you immunology a little bit, so you'll have fun tomorrow. But C-gas-sting signaling transcribes interferon response genes, okay? So it does that, analyze interferon response genes by just the virtue of what it is or the nature of what it is, will activate immune responses. Interferons are critical for immune responses, inflammatory responses. So the RNA itself can induce that C-gas-sting signaling. C-gas-sting signaling, and there are others of kind TLR8, TLR7, toll-like receptor 7 and 8. These signalings respond to the DNA fragments or RNA fragments that are just floating around in nucleoplasm or cytoplasm, okay, or otherwise. That's called double-strand RNA, DNA-based signaling events. These sort of signaling induces various kinds of immune responses, including T-cell activation or otherwise. T-cells are? How many of you know what is T-cell? Immune cells, but what do they do? Okay, we talked about T cells earlier. We talked about B cells earlier. We talked about antibodies earlier. T cells are your specific warriors that kill the foreign object, right? CD8 positive T cells. CD8 is a receptor on the cell surface of a specific subset of T cells that are essentially killer cells. So they're cytotoxic killer cells. They will secrete granzyme B, granzyme A, granzyme perforins. Perforins will make holes in the foreign object. They'll put in the granzyme A, granzyme B, they'll degrade things in, they'll kill them. What great killers, fighters we've got in our cells, in our body. How amazing. They specifically respond only to certain antigens. T cells are specific to specific antigens. We talked about antigens earlier, right? You guys remember?
Yes.
Antigens are the ones that are expressed by specific foreign objects or cancers. Cancers express specific antigens, okay? I always come down to cancer. But coronavirus had a specific antigen. So we had T cells specifically for those, right? And when you have antigens, you can use them to make what? Antibodies, but you can also make them to vaccines. That's right. You're so smart. I like that. Vaccines. We use vaccines, right? What with corona vaccine did you guys ever wonder? Okay.
Different types were there.
Good. What kinds of vaccine? What was it?
Whole virus vaccine, there was mRNA vaccine, there was
But
protein
what did they
vaccine.
do at the end of the day?
They stimulated this immune system to create antibodies, and they were
How do you
training
create
our
antibodies? Through antigens, right? mRNA, whole thing, whatever thing is just a modality to express antigens, to show your cells antigens so that the cells can make its own response against that antigen. Antigen is the word, okay? Antigens. Cool? It's not about how you do it. You can do mRNA, you can do protein, you can do whole viruses, attenuated one. There are all kinds of vaccines. But that's secondary. That's how you deliver them. But you actually deliver the antigen. That's what you're doing. You're delivering antigens so that your body can generate a CD8 positive T cell response against the foreign object so I can kill it. That's how cancer immunotherapies work. The guy who got 2018 Nobel Prize, who I told you yesterday, can improve lives of certain people by 10 years or more. People who've never got cancer again, they're cured of cancer, right? By immunotherapy. Immunotherapy is very powerful against doing lots and lots of things. Okay, you can think of all diseases potentially ones which are modulated by changes in immune system and how they can, how can you make vaccines? How can you make any kind of immunotherapy? People make antibody-based therapies. People make cell therapies. I make cell therapies in my lab. Things like that. Okay, cool. CAR T therapy. Who has heard of CAR T therapy? Okay, what is a CAR T therapy? You've spoken. Why don't you speak?
It's training your T cell for the by the chimeric antigen to the specific cancer.
Absolutely, thank you. CAR is a type of TCR. T cells work by TCR receptors. T cell receptor is TCR. A chimeric antigen receptor is similar to TCR. Essentially, helps respond to a certain antigenic thing. Okay, all right, cool. So that's how you make therapy. So see, all the way from immunology, I taught you from epigenetics, which has no meaning, and a cancer therapy, right? But why did we go there to begin with? When retroposons get expressed in situations that they shouldn't be expressed in, they'll make proteins that are foreign to the cell. They can make antigens of sorts. Epigenetic derepression creates neoantigens. New antigens. Okay? Neoantigens. New antigens are peptides that were not there normally. And cancer folks are using a lot of these neoantigens to make new cancer vaccines. In fact, Indian government has a huge program and huge amount of money committed to building new cancer vaccines, starting with cervical cancers, which happen all the time, right? Cool? A friend, very, very good friend of mine, my co-founder for GIAN Therapeutics called Satyet Shukla, he's an expert in cancer vaccines. He's made a vaccine that has gone on to be approved by FDA. Right? We could do a lot of things. New thing, new science. His USP is he uses nanopore. Somebody used nanopore the other day. Word. Nanopore is a sequencing technology that doesn't do short reads, that does long read. So he uses long read sequencing to discover neoantigens in cancers. And then he makes vaccines against them. How cool, right? Science, new technology, same problem, solving the problem, solving a clinical need, new ways to solve the clinical problem. Okay, cool. I'm just keep on saying this to you guys all the time. Think about it differently. Okay, and he's got lots of funding. He's got a lot of money to do this because people want cancer vaccines now, right? Would you want one? Right? If you got one, you'll be like, okay, if there was a universal cancer vaccine, would you take it? Okay, how many of you will take it? How many of you think vaccines are a piece of BS? Because lots of people in the world think that. Okay, lots of people don't believe that corona even existed. I'm not kidding. A guy on my plane from there to here, not this time, on a previous occasion, he was a Sardar. He was a he was sick, right? And he sat next to me. He was like, "What do you do?" I said, I'm a scientist. I do these things." Like, coronavirus was a thing propagated by pharma companies to sell products, sell vaccines. He didn't believe that corona existed. And I am not kidding. A lot of people out there who don't believe corona existed. How many of you don't believe corona existed? Coronavirus. Can you believe it? So you don't have to do science. You can be policy making and being in the community, generating awareness, cancer awareness. Be a advocate. Okay, be an advocate for diseases, finding new ways for diseases. You can do all of this. Career options, because you don't have to think that I have to go in the lab and do science. There's a lot more to this than that. We all need all kinds of people to solve diseases, right? For biomedical science betterment. Okay, think about that. But anyways, that was that was funny to me. I was like, "Are you serious?" Like, and he had lots of money. He used to he runs a business in California, and he's like, all of these people are out after my money. They want vaccines are not correct. Vaccines are bad. Corona didn't exist. So lots of people believe that, by the way. So I won't be surprised if people don't take cancer vaccines if there is one tomorrow, although we would have spent billions and trillions of dollars in developing one, and thousands and thousands and thousands of man hours developing it, lots and lots of people devoting their life to it, but there'll be lots of people who won't take it. So we need people who can voice for science, awareness. Our PM, our CM, our most influential people, politicians need that knowledge. They need awareness, right? Did I speak of this? Sorry, why am I saying that? I need to finish, but I will speak of something later on, which is funny. Okay, lines are great. Functions of lines in science. They are mutagenic effects. You guys get that? I just said that, right? They hop around. They'll create mutagens. They are normally silenced with epigenetic silencing. They're source of link RNAs, which I just explained to you. They can make long RNAs that can have function inside the cell. And finally, they can act as enhancers. How cool is that? They can actually act as enhancers. Whoa! So new enhancers, neo enhancers that are made in cell to do new transcription, and that's fun stuff, right? New pieces of biology one could study. Very new concept. Okay? All right? I want to talk 10, 15 minutes about, 15, 20 minutes about induced pluripotent cells, because I really like this stuff. I think this is one of the most powerful discoveries we have made that is and will have extreme influence as we go forward in regenerative therapies and therapies. Okay, what does induced pluripotent cell mean to you guys? What is a stem cell? Go ahead.
Means those cells that can differentiate
Good.
we are inducing some cells to gain that pluripotency that they can again get the ability to get redifferentiated and de-differentiated into.
Wonderful. Thank you so much. You guys can teach this class after this to your younger. Hey, how many of you really went on and asked you explained that to your parents the other day? You did? Good. Was it successful? Yeah, sort of. How was your experience? How about who else? Who else did that? Yeah, how did it go? Nothing. Try it again. Keep doing it. Keep at it. Keep at it. I have to keep the same thing, keep repeating the same thing to you guys, don't I? Right? Because we take time for longer term memory, responses, understanding, right? So it requires stimulation again and again and again and again to be better. Immune systems require multiple stimulations to become active. Your neurons require stimulations again and again. So keep doing it. Awareness is not easy, my friend. Making your society aware of things is not easy, because that's how human brains function, right? Induced pluripotent cells are induced pluripotent stem cells. You guys got this? Cool. These are two seminal papers from a guy called Shinya Yamanaka. I told you, Japanese scientist who made this happen. You should read stories about him. I don't have it here, but they went on it for years and years, thinking all about all combinations of things that will make induced pluripotent cells. Lots and lots and lots and lots of hard work. Two things. The top paper shows you they can do this in mice. The bottom paper shows you it can do it in humans. And that's it. That was just brilliant. I had finished my PhD by then. 2007, this paper came out. And I was like, wow, right? Wow. We can create stem cells from fibroblasts, from your skin cells, from your melanocytes, from anything in your body. How cool is that? That is the best, like that's one of the most powerful things you can do. You can create a stem cell. Why is that so powerful? Because then you can make them do whatever you want. You want to make them a liver, regenerative therapy, right? I can make him to a liver. I can make them to a neural cells, neurodegenerative diseases we just talked about, Alzheimer's, right? What happens in these? You lose your neurons. You lose your functions of the neurons. You can replace them, right? Beta cells, diabetes, type 1 diabetes patients are born without beta cells. They can't make insulin. Kids, and they require insulin all the time, right? Right? Can we transplant a functional beta cells in the pancreas of type 1 diabetes patients? Right? Very simple hypothesis. Just last month, a result came that they did this. They made iPSC. They used the iPSC cells, made them beta cells, put it in the patients, and patients, type 1 diabetes patients who were constantly on insulin, didn't need insulin for over a year. How cool is that, right? I can cure type 1 diabetes patients with my iPS-derived beta cells. I love this stuff. Santosh was doing this project in my lab, by the way, when he was there. We had just started. He'd done some things and then gone. So my idea is, can I epigenetically induce insulin through an insulin promoter change, epigenetic change, a permanent one, and I just can have insulin being expressed all the time from my beta cells. And I just transplant epigenetically edited focus beta cells. Can I use that? I still have the idea. Yeah? Can we do this? It would be so cool. Type 2 diabetes patients, if I just transplant this, I don't need to care about giving them insulin every day. Who's a family member with diabetes? How many of you guys see how many? If anybody's parents or whoever, right? Are they on insulin? My mother was on insulin pump, or the insulin pen before that. We do that every day, 3 times a day. Poke it, or they're on metformin, most of them, right? Metformin kills you internally, your other organs, right? So if we were to transplant a beta cells into the pancreas, would that create now insulin? If it did, so think about this. This is cool. Type 1 diabetes patients do not need insulin for a year. I mean, obviously the studies going on and all. The New England Journal of Medicine paper, New England Journal of Medicine is the most reputed journal in the world in medicine. No questions, okay? So the best research comes out there. Those research gets approved, clinically approved, and then becomes a therapy. I love it, right? Plus $100,000, that's a different issue, but at least there is a therapy available. Then your job will be to go and make that 100,000 into a 1,000 so your neighbors can actually take that, right? Cool? Think there's lots of things to do. But that has so much potential. You can do so many things. Blood cells, lots of people need blood cells. Remember hemophilia? We talked about the kids who require that. I don't know. We can make them into, factors that create the blood, that for those patients. Different organs, right? Of course, liver regeneration, all of that. Cardiacs, I don't know. So many opportunities, right? It's just brilliant. So the discovery to me blew my mind. At a young age, I was 26 years old, right? Just like you guys, a little older than you, but sure, blew my mind. I was like, wow. And it's all epigenetics, the differentiation, right? How did they do it? This is a data from that paper showing you different kinds of, this is a I think the colonies that essentially differentiate, these are iPS colonies, and they differentiate into different organs in vivo, and then they can do that. Okay? Very, very cool. This is done by overexpression of 4 transcription factors. 4 transcription factors. Cool. Yamanaka factors, named after him. He's become immortal. Doesn't matter what he did, he's become immortal. Doesn't matter his Nobel Prize or not. People will know these factors as Yamanaka factors forever. How cool. I am immortal because my name is immortal. I'm sorry, kidding. But yes, it is something, right? It's very nice. OCT4, SOX2, KLF4, CMYC. Wonderful, right? OCT4, SOX2, NANOG, 3 of them can do it alone together. So those are all transcription factors. Those are all transcription factors that do epigenetic reprogramming. Okay? So biggest use of epigenetics in creating new cells. All right? iPS cells are now being used to create cell therapies, immunotherapies for cancer patients. That's something I do in my lab too. I don't use iPS cells, but okay, we can use iPS cells too, right? That's nice, right? So I take your skin cells, I take it out, I grow it in a petri dish to make it an iPS cells, then I differentiate into whatever. I have a pool of that. Many of you, I don't know if any of you actually, you guys don't have kids, you guys are not even married. When you have your kids, at some point, they might want to save cord blood from you. Cord blood is when yes, umbilical cord blood. Umbilical cord bloods contain a lot of these pluripotent stem cells, and they can be reprogrammed to different things. Reliance made a business out of it. Reliance Life Sciences, it's called, just by storing the stem cells or cord blood cells from people. Very simple. Make money. You can make money in many ways. Okay? Good service to people who want to save their thing. Maybe it will be useful, maybe it won't. But really cool idea, right? Simple things, guys, make a lot of difference, can make differences. Okay, you should think about these things. That's what I meant. I like Swades because very simple ideas. Put a bulb somewhere, put a simple machine to be able to put the water, take the water wherever. Simple things. You don't need to go fancy. All right, we can do very fancy research. A lot of it can be very simply applied to a regular life and will have impact. Think about those things. Okay. So SOX2, NANOG, and POW F5, F1 is our OCT4. It's another name for it. We just scientists like to make things difficult, right? POW 5, F1. I was like, what the, right? I used a lot of this in my research early on. But anyways, SOX2, NANOG, this is on the left side is something called, do you guys see something? This is made in a specific way. So they work together, all 3 of them. They make a circuit. This is a circuit, okay? So that's sufficient because they can induce themselves by binding to an element called super enhancer. On the left side, I'm showing you elements that exist on the OCT4 promoter itself or OCT4 enhancers. You can see OCT4, SOX2, NANOG. They just bind next to each other and then recruit a bunch of transcription factors to change the cell phenotypes. All right? How cool. They're sufficient. The 3 or 4 of them are sufficient